As the name already defines everything in this interaction, that a gene which duplicates the phenotypic expression of the another gene. Okay, so a
Mhm.
duplicate gene interaction is considered as interallelic. So this is also an interallelic interaction, that interaction between two different genes, okay, or two different alleles of different gene. Interallelic interaction, because it involves the interaction between two different genes that essentially perform the same functions. So this is important, that two different gene has to do similar function, okay, meaning they are
Mhm.
not alleles of the same genes, okay. So these are not alleles of the same genes, but rather different gene at different loci with redundant functions, hence inter rather than intra-allelic. So it is like that. Suppose this is chromosome number 1 and this is chromosome number 2. Okay. So
Mhm.
there is a gene for hair color, for hair color. Okay. And at chromosome number 2, there is a gene that is Z and Z. So this is another gene Z, and this is gene H, and both gene
Mhm.
does the same function. Okay,
Mhm.
prepare pigment for your hair color. Okay, understood.
Yeah.
So but though both genes are two different genes at two different chromosome, or either at two different loci. This can be at chromosome number 1 but at different loci. Okay,
Mhm.
so there is a two condition, that these two different genes can be on two different chromosome or can be at different loci on the same chromosome. Okay,
Mhm.
meaning they are not alleles of the same gene, but rather different genes at different loci with redundant functions. Hence, not inter, that is, it is intra-allelic. So hence it is inter rather than intra-allelic. Understood? So this type of interaction is called interallelic interaction. Interallelic interaction. Duplicate gene interaction is a type of gene interaction that occurs when two genes produces the same phenotype. Two genes produces the same phenotype. This can happen when the dominant allele of both genes is present, or when the gene products of two loci have the same function. Okay.
Mhm.
When dominant allele, when dominant allele of both gene loci produces the same phenotype without a cumulative effect, in case the ratio becomes 15 is to 1 instead of 9 is to 3 is to 1. Duplicate gene interaction occurs in Shepherd purse plant. Okay, there is a plant named Shepherd purse plant. Okay. So now let's see one example of fruit shape in Capsula bursa plant. Capsula bursa. See, we will study about the fruit shape of Capsula bursa. Okay. See, two different images. Can you identify what is the shape of these two fruits?
Um, kind of triangular.
Triangular and
Um,
triangular and
circle.
elongated.
Okay.
Okay.
Mm-hmm.
And this is also known as Shepherd's purse plant. Okay.
Mm-hmm.
Its another name is Shepherd purse plant. Okay. We'll study about the fruit shape. Okay. That is two types of fruits, triangular and elongated. Triangular and elongated. So there is two types of genes, that is gene T and gene D, and both shows similar phenotypes, that is both is responsible for the fruit shape in the capsula bursa. So two types of gene we will see, that gene, or we can say allele, that is that controlling the fruit shape in capsula bursa, that is capital T and capital D as a dominant allele. Okay. So there are some conditions, that is if there is capital T and any other thing that is capital T or the dominant T or the recessive T, or there is a small d and a small d, you will get a triangular fruit. Okay. And if there is
Yeah.
small t or capital D with either a small or capital, you will again get a triangular fruit, because this D is also responsible for triangular shape in fruit, triangular shape of fruit in capsula burst. Okay.
Mm-hmm.
Again, there is a either this type of combination capital D with either dominant or recessive allele, or capital D with either dominant or recessive allele. There is no any modification or any type of change in the fruit shape. You will again get triangular fruit. Okay, and only if there is a condition of this, then you will get a elongated type. Okay,
Yeah.
so here you will get either small t or the capital T. Here you will get either a small d or capital D. Either there is a small d or a capital D. Okay, there is small t or a capital T. There is a small d or a capital D. Okay, you will get a
Mm-hmm.
triangular shaped fruit in all three conditions, and only in one condition you will get a elongated fruit. So this is the triangular fruit. Triangular fruit, and this is elongated. After today's session, I will provide you a worksheet, and uh, it is on either on Tuesday or Wednesday. So you have to do it after
Mm-hmm.
revising your sessions in genetics, and that test will be memory based test. Okay.
Okay.
After completing the test, you will text me and I will send you the answer key.
Okay.
Okay.
Mm-hmm.
So see, first for the first generation, the parent we take that is capital D, capital D, small d, and a small d. Okay.
Mm-hmm.
Small d and small d, and two different types of parent. Okay. And this will produce a triangular fruit. Can you tell me this is a pure line, pure genotype, or impure, or we can say that mixed genotype?
Um, it's pure.
Yes, pure, because triangular fruit is only dependent on either of the gene. Okay, if there is a
Mm-hmm.
capital T, then we can say that this is a pure one. Okay, or either it is a capital D, we can also say that this one is also a pure one. This is also a triangular. Now, can you tell me? I have given you a formula regarding to find the gamete that parent will produce. Can you recall that formula?
Uh, 2n.
2 to
Mm,
the power n. Okay. And what is this n?
the number of gametes.
Number of heterogametes. Number of heterozygous condition. Okay. Number of heterozygous
Mm-hmm.
condition. So in that case, can you tell me how many heterozygous condition can you observe that?
Uh,
Is there any heterozygous condition or not?
no, there is not.
Yes. If there is not, we will take it as zero. And then can you tell me what is the result of this?
One.
Yes, one. Only a one type of gamete you will see here. That is capital D and a small d and capital D. Okay. And
Mm-hmm.
what type of progeny is there? F1 progeny. It's all have a similar genotype. That is capital T, capital D, and small d. Okay.
Mm-hmm.
And its phenotype is triangular. Triangular fruit. The result is 100%. Okay.
Mm-hmm.
Now we will do F1 selfing. Okay. Self
Mm-hmm.
crossing. For that, we will take F1 progeny as parents. Now you will tell me what is the n value. Tell me.
Um, two
2 to the power?
of four,
Not
or is
4,
it
only
two?
2.
Okay.
Okay.
Mm-hmm.
Because for now, we are just observing that how many types of gamete this parent will produce, and after that, we will see that how many types of gamete this parent will produce. Okay. So there is only two heterozygous condition. This is this one and this one. Okay. So 2 to the power 2 is
Mm-hmm.
4. Okay. So this parent will produce four types of gamete. Okay. Can you tell me
Mm-hmm.
what are the combination of gametic genotype?
Um, T, uppercase T, big T, um, big D, big D,
No, tell me again. I will write it down.
big T, small T.
No. To do that, you have to first, if you are facing such type of problem, first you have to just separate all the alleles. Okay. Separate all the alleles. Okay. After separating all the alleles, you have to make a possible combination. So these are one set of alleles and these are another set of alleles. So this one combines with this to produce capital D, capital D, and again this one combines with this, that is capital T and a small t. After that, this will combine with this, and again this will pair with this T. I think now you get it.
Mm-hmm.
Do you?
Yeah.
Okay, so this one is a pair of allele and this one is another pair of allele. Okay.
Mm-hmm.
Similarly, you will get capital T with this D, capital T with this D, small t with this one, and this small t with this one. Okay.
Mm-hmm.
So when you are going to prepare the number of gametes or the possible combination of gametes, first separate all the alleles. Okay. And after that, after then you can make the possible combinations of gametic pair. Okay. Understood or not?
Yes.
Okay. Now, after that, when we draw the Punnett square, so now let's make the genotypic combination. Tell me what is the genotypic combination for this progeny?
Uppercase T, uppercase T, uppercase D, uppercase D.
Yes.
Um,
Okay.
big T, big T, big D, small d.
Yes.
Big D, big T, small t. Big D, big D. Big T, small t. Big D, small d.
Here,
Big T, big T, big D, small d.
here,
Big T, small t, big D, small d.
yes. No, here,
Um, small t, big D, small d.
here.
Small t, small t, small d, small d.
Yes. No, I'm writing all these things. You just check it. I'm writing correctly or not. Okay.
Mhm.
So this one makes what type of fruit?
Which one? Oh,
First,
triangular,
triangular. And this one,
triangle,
this one,
triangle as well.
this one.
Triangle.
Okay. So can you tell me what is the ratio of triangular fruit is to elongated? Count that. Tell me the ratio of triangular fruit is to elongated fruit.
15 over 1.
Yes. 15 over 1. So all these are triangular one. Okay.
Mm-hmm.
All these are triangular one. And only this one is the elongated. Okay. So here again we what we see that we have a different ratio as Mendel suggested in F2 generation of the progeny. Okay, but the
Mm-hmm.
number of progeny is same. Okay, we always get 15 different types of genotype, but 16 different types of genotype. Okay, genotypic ratio is always same, but there is a difference only in the phenotypic ratio. Hello.
Yeah.
Okay, let's check it that about the genotypic ratio. Is it always 9 is to 3 is to 3 is to 1? That is 12, 24, 12, 12, 1 or not? Okay,
Mm-hmm.
can you do that for me? Make the genotypic ratio.
Um,
Capital D, how many we have this genotype?
uh, 1,
It is there. Yes, 1. And capital D, here is the 1. And
2,
the next, here, 2. And the next one is this one. And again, 1,
2.
2. We have 2.
Yeah.
1, 2, 3,
4.
and 4. Okay.
Mm-hmm.
Again, 1,
1, 2.
this is the one, and next, only 1.
Yep.
Okay. Capital T, small d, and this one, 1,
2.
2. Next, this one, small d, small t, capital D.
1,
1 only. Okay. Next,
2.
2. So if we arrange that, we can get 1, 2, and this one is here, 4. And this one is okay. 12, 24, 12, 12, 1. Okay. So
Mm-hmm.
genotypic ratio is always same in any type of F2 generation. When we study about the dihybrid cross, the genotypic ratio is always similar. That is 12, 24, 12, 12, 1. Okay.
Yeah.
But there is a difference in the phenotypic ratio. That's why we call it a variation from Mendelism. Okay.
Mm-hmm.
Any problem?
No.